Thank you all for coming. We have a marvellous turnout here this weekend, and the keynote speaker for this conference is Dr. Alan Cantwell, and I'm sure you've all read the bio which is online and in the program, so without further ado, here is Dr. Alan Cantwell.
Thank you very much. I'm delighted to be here, and it seems strange that my connection with Trevor was sarcoidosis, and I was always interested in sarcoidosis, and through my research, Trevor found me, I found Trevor, and I've been retired from medicine now for 12 years, and Trevor single-handedly has resurrected my past career. So today we're going to talk about the cancer microbe and the Russell body. There's a few things that we should know about the cancer microbe. First of all, it's taboo and heresy, and nobody believes much of what I have to say, but nevertheless, the cancer microbe basically is a generic term. It has to do with microbes that can be found not only in certain cancers, but in many different types of diseases. The bacteria that have been grown from cancer have been basically simple bacteria, such as staphylococci, streptococci, what they call Corynebacteria. So these are very simple germs that doctors don't pay much attention to, and ordinarily they consider them secondary invaders or contaminants of no great significance. The cancer microbe in tissue appears as small round forms as well as larger forms which are known to pathologists as Russell bodies. And again, I will show you slides to show you evidence of this. The cancer microbe lives within the cell and outside of the cell. It's an intracellular and extracellular microbe. Elements of these microbes are also found in the blood, which, as some of you know, also contain elements suggestive of bacteria, even though the medical profession basically states that the blood is sterile. But I think that the origin of these microbes is basically from the blood. The cancer microbe in tissue is best detected by using a special stain called the acid-fast stain. The acid-fast stain is a tissue stain that has been traditionally used to detect mycobacteria. Mycobacteria are a certain type of bacteria that produce diseases like tuberculosis and diseases like leprosy. Unfortunately, the routine stain that the pathologist uses is not very good to detect these bacteria, so staining is crucial. Now, there is no question that these cancer microbes live in the body in the cell wall deficient form. And cell wall deficient forms have all kinds of names that scientists and microbiologists have put on them, such as L forms, mycoplasma, so-called large bodies, Russell bodies, and so on and so forth. By definition, cell wall deficient bacteria are pleomorphic, meaning they exist in tissue in more than one form. However, the primary form of these microbes in tissue is the round coccus form. The reason that they are pleomorphic in tissue is that some of these round forms are very, very tiny. It would be like you were comparing a marble to a ping pong ball, to a baseball, and to a basketball. Each of those forms are round, but collectively they're different in terms of a marble is a heck of a lot smaller than a basketball, and I'm going to show you the way these germs live in the tissue in chronic disease as well as in cancer. The other common form of bacteria, of course, is the rod form or the stick form, which would be like comparing a baseball to a baseball bat. The baseball bat representing the rod form. But rod forms in tissue are quite rare, and I'll show you some of those. According to cancer microbe researchers, and I'm just like one of the last cancer microbe researchers, so that all the stuff that I'm showing you, I basically learned from other cancer microbe researchers, all of which who at this point are dead. So I'm sort of the last of the Mohicans, and I'm sort of hoping that some of you people here, particularly you younger folks and particularly physicians, will pick up on this work, because my feeling is that you cannot understand cancer and you cannot understand chronic disease unless you understand these bacteria. Cancer microbe research, believe it or not, basically extends back into the late 19th century, when pathologist William Russell first described the parasite of cancer. And as I said, rule number 9 is medical science doesn't believe a word of this. So welcome to the twilight zone of medical science. Here is William Russell, who died in 1940 when I was only 6 years old. And he first found these microbes in all kinds of cancer that he examined, and he found the microbe within the cell, outside of the cell, and he said that these microbes were barely visible under the microscope, but they could be as large as red blood cells, which are about 6 or 7 micron in diameter. And Russell also found these so-called parasites in tuberculosis and in syphilis and in skin ulcers. Well, needless to say, within a decade or two, the bacterial or the parasitic theory of cancer was totally thrown out around the 1910s, when they realized that there was no such thing as bacteria in cancer, that cancer was not an infectious disease, cancer was not a contagious disease, and anybody that continued to study bacteria and cancer was sort of out of their mind. So here we see an example of what I consider Russell bodies. And don't forget, pathologists do accept the fact that there are Russell bodies. It's just that they don't accept the fact that they are microbial. Most pathologists seem to agree that Russell bodies are immunoglobulins of sorts. But this particular slide was taken from a teenage Mexican-American girl who died of Hodgkin's disease. And you can see on the right a cell that has these forms, and on the left you can see the forms outside the body. Now, this is taken with a special acid-fast stain like you would use to detect tuberculosis or leprosy. And you must also, when you look for these germs, you must also use the highest power of the microscope, and you must use oil immersion, which allows these forms to be seen at a magnification of 1,000 times. Now, first of all, when it comes to cancer, most pathologists are not going to use a special stain, and they're not going to put oil on the slide, because that's just not what they do. But in order to look for bacteria, you have to have the highest power, you have to use oil, you have to study it carefully. And the differences between swimming and scuba diving. You know, if you got goggles on, you can see all the fish. If you just got nothing on, you're not going to see anything. So this is the key, is the special stain and the magnification.
Is this bright field or dark field?
No, this is regular. This is a regular light microscope. All this stuff is regular microscope, and all of this stuff is at 1,000 degree magnification, except for one slide in which I'll show you that. Now, here is Hodgkin's disease of the lymph node. And again, you can see on the top, you can see the larger Russell bodies, and you can see these smaller coccoid forms. Now, those tiny coccoid forms down here, those are about the size of staphylococci, common ordinary staphylococcus. But these larger forms here are 3 or 4 or 5, 6 times as large. And this happens to be a Gram stain, which is the traditional stain to show bacteria. But unfortunately, the Gram stain, the traditional stain for bacteria, does not show cell wall deficient bacteria very well. Now here we have a giant, really giant Russell body. Now I'm going to show you that most pathologists would certainly not would not consider that to be a germ. In fact, the size is so large that it might make you suspect that you were dealing with a larger fungus. Now, people who have studied in depth the germ that causes tuberculosis, the mycobacteria that causes tuberculosis, have found that the tuberculosis germ, like a lot of germs, has a very complex way of multiplying. Ordinarily, doctors think that, well, a germ is a germ is a germ, and a germ divides in half, and you got 2 halves that look just like the parent germ. And that's called the monomorphic theory of microbiology. But people who have studied bacteria in a different way realize that the life cycle, or the way bacteria reproduce, is very complex. Now, this is taken from a book. I have a copy here if you'd like to see it, by a Spanish physician and microbiologist. And he studied cell wall deficient forms of tuberculosis, of the tuberculosis germ. And he found, using a phase microscope, that the germ can go all the way from little tiny little dots, the size of staphylococci or the size of granules, up to the size of very, very, very large forms here. Now, those large forms are comparable to Russell bodies. Here is another picture from Zalabarder's book. Zalabarder died a number of years ago, and his work has been largely forgotten. But this is the kind of thing that you have to sort of understand the variability of these cell wall deficient bacteria in order to be able to recognize them in tissue. After all, if you're going to go hunting for tigers, you've got to know what tigers look like. And if you're going to be hunting for bacteria in chronic disease, you have to know what they look like. And you have to know how to color them, and you have to know the magnification in which to find them, and you have to have the patience. You might have to spend in certain diseases a couple of hours before you start to see what, to me, at this point, is pretty obvious. But for the first 10 or 15 years of my career, I was seeing this stuff all the time, but I didn't identify it. It's like Sherlock Holmes says, you see, but you do not observe. And there's a big difference between that, It's plus, sometimes the hardest things to see are the things that are right in front of your face. I'm sure we all know that's true. Now, here is lupus erythematosus, which is one of your Th1 diseases. And here is a very, very, very large acid-fast, in terms of its staining red-purple, Russell body. Now, I don't think anybody but me has found Russell bodies in lupus, and most pathologists say, you got some junk on your slide. But, you know, if you look at a lot of different diseases using these special stains, after a while you start to see a commonality that you did not know exist. Now, here again is the same slide from lupus, and on the right, you will see this is the common way the germ looks. It's those little tiny, little coccoid, little staphylococcus looking forms here. And this is the skin of lupus. Now, when this particular lesion was cultured, my microbiologist friend Dan Kelso grew Propionibacterium acnes. Well, that's a common germ that is on the skin and that nobody pays much attention to because it's considered an ordinary contaminant. But my point is that if you look at, this is what the germ looks like. Now, if you superimpose that over here, it's very similar. In other words, the little tiny coccoforms, and Propionibacterium acnes is basically rods and cocci, you will see that there is a similarity between what you see in the tissue, and this is a special stain, and what you grew. And I'll show you more and more of this as we go by. And again, from lupus, here's the microbe in tissue, and here is the microbe in culture. Well, people can say, well, this germ is supposed to have rod forms, supposed to have stick-like forms. Why are there no stick-like forms here? It's because when a germ like this infects the tissue, the rod forms disappear, and the form that's infective is the round form. And again, you have to understand this is all heresy. Okay, now this is the disease that started my career. A disease called scleroderma, hardening of the skin, a collagen disease, a connective tissue disease, an autoimmune disease. And back in the 60s, I found, with the help of a technician, acid-fast bacteria in scleroderma. And everybody knows that scleroderma is a disease of which there is no known cause and no microbial etiology. Here are the bacteria. Now, when you find these bacteria are definitely rod-shaped. You can see the little stick forms. And they are acid-fast in terms of they stain red to purple-red. And these germs are a dead ringer for the kind of germs that you see in tuberculosis and leprosy. Well, I thought I was the only person on the planet that saw this, and then about a year later, one of my colleagues in dermatology said, well, there's this woman in San Diego. She says the same thing you do. Scleroderma is caused by these TB kind of germs. And then he went on to say that now she thinks that these TB germs are involved in cancer. Well, that was a little too much for my brain to handle. But nevertheless, I met her, Virginia Livingston, MD. She died in 1990. She was a dear friend. Everything I know I learned from her and her women colleagues. However, the common form in scleroderma is the coccus form. And here you can see the collagen, and in between the collagen bundles are these coccoid forms. This happens to be a Giemsa stain, which is another stain which can be very helpful to show these germs. Giemsa stain is traditionally used to demonstrate malarial parasites in the blood. So anyway, in scleroderma, you have acid-fast bacteria, like I showed you, which are very, very rare. If you see acid-fast bacteria in scleroderma tissue, it's like finding a four-leaf clover. And you have to, I have looked for hours and hours and hours to find and to demonstrate acid-fast bacteria in scleroderma. But this is the common form. And from this disease, eventually I went on into sarcoid and into cancer, and from the same disease, Virginia Livingston independently went on to become the leading promoter of the cancer microbe theory in the last half of the last century. Now here's sarcoidosis of the skin, and you can see in the center here these coccoid forms. This is within the granuloma area of sarcoid. Here is sarcoidosis of the lymph node. It's a Giemsa stain. You can see these tiny little coccoid forms. The cancer microbe has a tremendous affinity for the connective tissue. So it's interesting when you study the nodes, kind of the best place to find them is actually around the nodes rather than in the nodes. And of course, a lot of this stuff has been published in the medical literature, and you can find examples of this work on the PubMed website. Now, here we get into cancer. I mean, actually, I never wanted to get into cancer because I never wanted to be known as a quack like my friend Virginia Livingston. But after I started studying sarcoid, I realized that, and I didn't know this, that some cancer patients, when the cancer spreads to the lymph nodes, sometimes the lymph nodes can look exactly like sarcoidosis. So in some ways, sarcoidosis is midway between an inflammatory disease and a cancerous disease. And so I eventually decided, what the heck? And so I started to study cancer in the same way, looking for the same type of germ that we found in scleroderma and in sarcoid. And sure enough, here is mycosis fungoides, which is a T-cell lymphoma. And here is a picture of the lymph node showing these forms in the lymph node of a lymphoma. Now, we also studied some patients at autopsy. I was told by Florence Seibert, one of the fabulous scientists of the last century who got involved with the cancer microbe work, along with Virginia Livingston and Eleanor Alexander Jackson and Irene Diller, whose scientific works are just fantastic. And Florence said, well, Alan, you can't just, show this stuff in skin. If you think that these microbes are involved in a disease, then you've got to show them in other organs, which is how I started to get involved with autopsies to some extent into lymph nodes, lungs. And fortunately, I was at Kaiser where we had easy access, to this. I was really blessed to be at Kaiser where everything's under one roof. And even though the pathologists, I think, kind of looked at me kind of strangely, they were cooperative and said, okay, if you want a lung, you can, we'll get you the lung and we'll stain it and you can look at the lung or what have you. So I learned a lot by looking for these germs in autopsies, which is, the autopsy is the ultimate diagnosis. And so one of the mycosis fungoides cases was autopsied. And here you can see these coccoid forms in the heart muscle. Well, in this particular case, the heart was normal. But in other cases, it's not normal. I'm not sure that pathologists pay any attention to this stuff. And, there's some question now about arteriosclerosis being possibly, there might be microbes involved. And I personally think that the heart, after all, if you're a microbe and you want to live and you want to live in blood, heart's a great place to live. This is breast cancer. And again, this gets kind of far afield from dermatology, but I had a woman who developed skin metastases from the breast to the skin, which is not terribly common. And so I decided, well, let's take the skin and look for these cancer microbes in the metastasis. And of course, we found it, and we grew a Staphylococcus epidermidis type germ from her skin lesion, which ordinarily would be, well, it's a contaminant, it doesn't mean a damn thing. Well, we went back, and the pathologist was able to get her original breast cancer surgical specimen biopsy. And when you looked at the breast cancer, there is old Russell again with his intracellular microbes. So again, you could say, well, this is the parasite of cancer in breast cancer, and you can see this is the Staphylococcus. There are some strange-looking little granules here, and if you look closely, you will see some spicules. Now, Virginia Livingston said that was kind of an unusual finding, but she talked about that when she grew her cancer germ. She said, Alan, you find these granules, and these granules have these spicules, which again is, to our mind, an indication that these cancer germs are very closely related to the TB germs. Or you could say that the TB germs are very closely related to the cancer germs. So, and again, these are acid-fast stains. These are examined under oil and at a magnification of 1,000 times. And again, here's the breast cancer, and here's the Staphylococcus. And if you superimpose this on this, they're dead ringers for one another. Here is, again, a particular cell from breast cancer. And if you look closely, you can almost see that they're almost like little fungal hyphae coming out of that cell. You don't see that very often, but I think that's going to equate to some of these pictures of blood cells where you see these little fungal-like hyphae coming out of the blood. So again, this shows, in my view, an intracellular parasite. And again, I think most pathologists would kind of scratch their head and leave the room, which they have done. And again, here's another cell from breast cancer. You can see some of the larger coccoid forms at the bottom there. And again, one final view of the bacteria that was cultured from the skin metastases, and showing you that cancer microbes basically are very simple little microbes that nobody pays any attention to. But these are the microbes that have existed with us and on us since time immemorial. I mean, that's why surgeons scrub their hands, put on gloves and wear masks, because you don't want these little simple germs to get in your body when you're being operated on. Now here's lung cancer. I haven't studied lung cancer. This is like a rare example, because dermatologists shouldn't be studying lung cancer. But again, you see a cell here is almost identical to what you saw in breast cancer. Now that doesn't mean that smoking does not cause cancer, but what smoking may do is damage your lungs and allow these germs, which we all carry. And that's another problem with cancer microbes. Everybody carries cancer microbes. And doctors have a hard time, pinning the rap on a germ that we all carry, because the idea is healthy people should carry, a bad germ, but, sick people should carry the germ that causes it, but healthy people shouldn't. And that is not the way the cancer microbe works. Everybody has it. Now, when my partner developed prostate cancer 2 years ago, I thought, well, gee, I always wanted to study prostate cancer, but prostate cancer is far afield from dermatology. But I thought, well, this is make it or break it kind of thing. Maybe I am crazy. Maybe maybe I am wrong that there's no such thing as a cancer microbe. Blah blah blah. So I asked the pathologist, and I'd been retired for a number of years, and I asked her if she would please do an acid-fast section, which she did. And this is prostate cancer, and I will show you. This is some more prostate cancer. I mean, there's some pretty big little coccoid forms there. And again, you see intracellular forms, and here you see the clumped forms, and here you see the extracellular coccoid forms. This is Russell's parasite of cancer that he talked about. This is the 150th year of its birth. I think that's quite amazing. But how do you get people interested in this? I mean, this is like every man's worst nightmare. I got to take my prostate out. That's the end of my love life, true. But there's like no interest in this. I tried to get a few of the doctors interested in it, and they're not. So, don't ask why, because it's X-Files stuff. Anyway, I wrote my first book about this in 1990, and I talked about the history of the cancer microbe, and I have some copies if you want to buy some. I'll have to get them out of the car. Then last year, to celebrate Dr. Livingston's, it'll be her 100th birthday this December, as well as to honor some of the other three women who I think are, like the greatest scientists of the last century. I wrote this book in conjunction with the Cancer Federation, who wanted to celebrate her life and her achievements. And so I wrote a small book called Four Women Against Cancer. The cover, again, is that teenage girl that died of Hodgkin's disease. And this is the microbe at autopsy in her connective tissue. And this is my dear friend, Virginia Livingston. It was taken around 1980. She had a clinic in San Diego. It closed a couple of years ago. Fabulous scientist. If you get an opportunity to read her books, you will learn an awful lot from her, as well as her women colleagues. Now, fortunately, a lot of this stuff, is preserved now on the internet, thanks to Trevor, who has really, as far as I'm concerned, resurrected this work. And I'm glad that it's complementary to his ideas. So it's a great marriage. I don't quite understand this stuff, but I'm like you. I'm learning, and I'm just amazed that that vitamin D could possibly be involved in healing. And so there are papers on the website. We're going more into detail about Hodgkin's disease and into scleroderma, into skin cancer, which I did not discuss. Skin cancer is like so easy to see these germs in skin cancer. It's unbelievable that it's not seen. Again, obviously, the sun, does something. But again, I think you will find that the microbe is there. And there's papers on sarcoidosis, and also a paper on sweat glands, because I have a feeling that some of the entry, at least in the skin of these microbes from the blood, is via the sweat glands. So if you read these papers, and don't just read my stuff, if you have an opportunity, read the work of previous scientists and researchers upon whom, my work is based. I mean, this is not my work. I have approached it, in a way perhaps different than most of the other cancer microbe researchers. But it's basically, one scientist growing on another scientist and so on and so forth. So I hope you'll have the opportunity to study this as well as the references very, very carefully. And of course, in the next lecture, we're going to talk about the cancer microbe in AIDS. So I think that's really very, very exciting work, and So stay close. Thank you.